Okay, well, welcome everybody to this morning's Innovation Showcase. We are delighted to be hosting this session, and it's a great opportunity to showcase some of the amazing work that is happening in life sciences. All of the companies that are presenting today are actually finalists of our Bio News Round Awards. So if you haven't already, I will pop a link into the chat box so that you can vote and you can check out some of the other companies. Just before I hand you over to our first presenter, if you don't mind just keeping your microphones on mute throughout the presentation, that would be brilliant. However, we do like these sessions to be interactive, so please feel free to put any questions or comments in the chat box. We do have closed captions enabled as well if you do require that accessibility. And I think that might be it from me. Oh, just to mention as well, we are recording this session and that will be available after. So with that, I will hand you over to Pooja, who is our first presenter from Astex Pharmaceuticals, and I hope you all enjoy the session.
Thank you. So I'll attempt to share my presentation. Can you see that in full mode?
Yeah, that looks good.
Okay, and I have laser pointer. Hopefully you can see that as well.
Yeah.
Yeah. Okay, great. Okay, thank you. So I just want to thank the organizers for giving me the opportunity to speak today. My name is Pooja Pathurian, and I'm a director at Astex Pharmaceuticals. So I'll take you through a bit of an overview about the company and kind of go into what we're doing in terms of innovation to elaborate our platform at Astex. So basically, we're a global developer of small molecule drugs for unmet medical needs, and we're looking at oncology and CNS therapeutics development. So the company started back in 1999, and it was founded by three people. One was Dr. Haran Jyoti, who's now our CEO and president of Astex Pharmaceuticals in the UK. And the other founders were Sir Thomas Blundell and Professor Chris Abel as well. And they found the company and pioneered fragment-based drug discovery and discovered the Pyramid platform, which I will talk about in my upcoming slides. So basically in 2011, we merged with an American company called Supergen. And then in 2013, we were acquired by Otsuka Pharmaceuticals, which is a Japanese company. So now we're part of the Otsuka family. So it's actually our 10-year anniversary being part of the Otsuka family. So we had quite a few celebrations this year. But it's actually expanded the 20 years we've been at Astex. It's got us into more of the CNS side of drug development. So that's what we're looking at the moment in conjunction with all the oncology work. So we're based in the UK, and the UK site is about 150 employees. It's in Cambridge, and we're doing mainly the discovery research and preclinical development. And our US site is located in Pleasanton, California, and this has about 130 employees, and it's more on the clinical development and regulatory side. Something we quite pride ourselves on is our strong partnerships with both pharma and academia. We have partnerships with AstraZeneca, Janssen, GSK, Novartis, Merck on the pharma side. And in academia, we have collaborations with ICR, Sierra UK, NKI, and Newcastle. And this is just to name a few. And I can't stress how important it is to have these partnerships. It has actually led to two of our marketed drugs. One is Kisqali, and this was FDA approved back in 2017. And this was a collaboration with Novartis on a CDK4/6 inhibitor. And it's now in patients with advanced breast cancer. And then in 2019, we had our second drug approved, which is Balversa. And this is an FGFR inhibitor to treat metastatic and advanced urothelial cancer. And this was a collaboration with Newcastle University and Janssen. And we also have ongoing collaborations with our Japanese colleagues at Otsuka and Taiho. One of them is a KRAS collaboration with MSD, and this includes our Taiho colleagues. And we also have a SHP2 program with Taiho that has been licensed to MSD. So then I'll go into our bread and butter of what we do at Astex. So The company was based and discovered on fragment-based drug discovery, and I just want to discuss like why we decided to start doing this and why we invested in this technique. So as most of you know, most companies screen by HTS. They typically screen thousands and thousands of compounds in the micromolar affinity, and they're usually in the 350 to 500 Dalton range. What we're doing is screening fragment hits. So they tend to be very weak. They're in the millimolar affinity, and they're between 100 and 200 Daltons. So the advantages of fragment-based drug discovery is we have an optimal fitting. You got smaller compounds that lead to smaller lead compounds. They have good ligand efficiency. One of the drawbacks is obviously we're dealing with very low affinity molecules. We need to have high sensitive techniques to detect hits. So we do this by X-ray screening, which I'll discuss in the next slide. The reason why we do fragment-based is we can sample the binding space a bit better. We can also find novel binding sites on the protein. And we've actually discovered quite a few targets that we can apply this method to, and this includes kinases, protein-protein complexes, and allosteric modulators. And this technique has been used over the last 20 years, and we now have proven this technique to be quite useful, and we have clinical candidates that have been identified by this approach. So this is just showing how we had a fragment and how we tried to optimize and grow into a binding pocket. So this is our Pyramid platform. So I'll just go through this with you. So we have our fragment library, and just to give you an idea, we're now on our version 5 of our core fragment set. So it has evolved over the last 20 or so years. We have more than 2,000 compounds in this fragment library set. And a subset of that library is our X-ray set, and that's the set that actually goes directly into our crystal system. And the rest of the core fragment library is screened by other biophysical techniques. We also look at targeted and virtual screening sets as well. So typically when we start with a target, we will set up a crystal system, make sure it's robust, it's soakable with tool compounds. And then when it's ready, we will screen our X-ray set. And we typically use both in-house and also synchrotron radiation to collect data. We also look at other biophysical techniques to screen the whole core fragment set, and that includes NMR and SPR and thermal shift. And we also look at ITC to do KD determinations at the initial stages. Obviously, because we're using very weak fragments, we don't get very much KD determinations at that stage, depending on how weak or strong the hits are. And now we're involving the platform to include cryo-EM, which I'll talk about in the next few slides, just to say how we're expanding the platform, and it's actually giving us access to different protein targets. So once we've done our screening through the Pyramid platform, we will identify hits, and then the chemistry team will get involved and we'll do structure-led optimization. And basically what they'll do is they'll add different functional groups to our fragment hits, expand it so they can find optimal interactions with the binding pocket, and they'll go through several iterations until we have a candidate we can use in preclinical work. So how are we expanding this platform? We're also looking at informatics, machine learning, AI. So some of the things we're doing is we're looking at image recognition of crystal detection and crystallization wells for our screening. We're also learning from the output of our previous screening campaigns, and we're using public domain tools such as AlphaFold. We're also trying to improve our automation data processing analysis, as for both X-ray and cryo-EM. We're improving our computation methodology by combining AI and QM. And later on in the more advanced stage, we're looking at generative models for de novo design, models for physical and DMPK properties. We're also looking at physics-based methods. And the chemistry team is looking at ways that predict reactivity and synthetic routes, so we can basically generate compounds a lot more quicker than we used to. And this is going to be my last slide. It's just going to mention what we're doing in the cryo-EM space. So we now have our own cryo-EM facilities based at our UK sites, and it's housing 3 microscopes. One's our cryos, and then we also have 2 glaciers on site. And we've heavily invested in this technique. About almost 7 years ago, we invested in this technique. And it was just to open up the protein classes we don't have access to using X-ray. So we're looking at multi-protein complexes, including integral membrane proteins and ion channels. So difficult complexes and proteins that we can't normally generate by X-ray, we're now looking at in cryo-EM. And we're now building our experience from what we learned from X-ray and streamlining it for EM, so we can try and make it a high throughput method. Also, it'll take some time before it gets to the X-ray stage, but we are generating maps that are in the 2 angstrom range. So it is getting close to X-ray resolution, or at X-ray resolution. It's now just trying to speed up the data collection and processing. And when we first started our investment in cryo-EM, we did it on proof of concept studies. So we looked at established targets. We looked in-house. One was PKM2, and you're welcome to obviously look at that publication. But now we're using cryo-EM on live targets. And then another thing that has been really useful is the Cambridge Cryo-EM Pharma Consortium. And this was founded back in 2016 with Richard Henderson at MRC. And we have both our academic and pharma partners in it. So that includes University of Cambridge, Thermo Fisher, AstraZeneca, GSK, Heptares, and UCB as well. And I'll stop there. So just thank you for listening. And hopefully that gave you a bit of a snapshot of what Astex is all about.
Thank you so much, Pooja. That was a really interesting presentation. Just before we move on to the next one, we do have some time for questions if anybody has some for Pooja.
Yes, thanks, Pooja. This is Marshall. My question is, you have 2 approved drug, and it's approved FDA. What's your strategy for the other market, like Europe and also China? Because I know you are part of the Japanese company, pharmaceutical, but whether your company has the independence, to negotiate regional deals, license deals.
Yeah, it's a good question. It's probably a bit out of my space, if I'm honest. So yeah, it's something we'll have to think about when we especially go into CNS and therapeutics, because Otsuka is obviously a Japanese-based company. Right now we are focusing on the Europe and the American market, but we're also looking at other countries as well, because obviously there's unmet need in other countries. So we're starting to do that a bit more. We get a lot of input from our US colleagues who are doing the more clinical side of work.
Okay, thank you.
Thank you, Marshall. Do we have any more questions at all from the audience? No? Okay, well, thank you so much, Pooja. I will bring up next. We have Helena Alexic from Pharmenable. Over to you.
Hopefully you can see that. So yeah, thank you very much for having us today. I'm very happy to be here to tell you a bit about our work at Pharmenable. So to give you a summary of what the company is all about, so our mission is to develop targeted oral therapies that are highly effective and with fewer side effects for diseases with a high clinical need. We were kind of, back in the day, spin out from Cambridge originally, from the Department of Chemistry, looking at methodologies around diversity generation and how to build up kind of novel, 3-dimensional, diverse molecules in a small set of synthesis steps. And that's still something at the core of our technology and what we do. And we're focused on developing the next generation of small molecule drugs with these properties in mind. So focusing on really tapping into that novel chemical space, coming up with IP-free molecules, using 3-dimensionality and complexity to reduce side effects and to come up with small molecules that really target things very specifically. And also delivering chemical diversity. So really tapping into the kind of full potential of the chemical space that's out there. So at the moment, we have our own pipeline of drugs. So we've got 2 discovery stage programs in oncology, and we're also working with partners across disease areas. So, for example, we've had a kind of long-standing partnership with, say, Heptares, originally in neurology. We're now looking at pain indications as well. We're also working with Denali Therapeutics in neurodegenerative. And we have this amazing platform that's disease agnostic and target family agnostic. So we're always looking for opportunities to kind of apply it to interesting targets in disease areas. And in terms of what's kind of really specific about our molecules, so firstly, as I mentioned, we're very much focused on chemical diversity, so delivering novel, diverse scaffolds and chemotypes that are also modifiable, and make a really good kind of starting point for the drug discovery process. So we find this is really helpful in a number of scenarios. So, for example, if you have a target that's just been kind of very tricky to unlock in other ways, if selectivity has been a particular issue for the targets, and also in instances where you need a very specific balance of properties. So we really specialize in kind of navigating chemical space and finding the right molecule with the right properties for the therapeutic area in question. Now, in terms of navigating normal chemical space, we have this kind of dynamic approach. So it's not a kind of static virtual library. Instead, we model the targets, and then we dynamically generate virtual molecules that are specific to that target. So it's kind of a bespoke per target approach. And we also focus on novelty, so it's IP-free from the start as well. We build in three-dimensionality and complexity as required by the needs of the target and the project. And finally, we're very focused on synthetic accessibility. So we have an average of 5 synthetic steps, and across all of our programs and all of our compounds we've ever made, we've had a synthesis success rate of over 90%, which is fantastic, because we're talking about kind of novel compounds in de novo design here. Just to tell you a bit about who we are, we've been a remote first company from the start, so we've got people in a few different countries, but we're very kind of Cambridge centric. Just maybe a lot of familiar faces in this slide. We recently raised our second funding round, which was 7.5 million. So that's our piece of good news in the Good News Awards. And as part of that, so Jeff Moore from MP Healthcare, which is the venture arm of Mitsubishi Pharma, joined our board. And we also recently had Keith Blundy joining, who's formerly Storm and CRT. And something that's special about our team is that we're very interdisciplinary in our approach. So we have medicinal chemists and computational chemists working very closely together to come up with the most kind of innovative methods and small molecules that they can. And to tell you a bit about our approach, so if you imagine the possible chemical space of all small molecules that could one day become drugs. So that chemical space is astronomic. It's estimated to be 10 to the 62. And generally speaking, existing screening collections only focus in on relatively small areas of that chemical space, and often tend to be somewhat two-dimensional in nature as well. What we focus on is navigating the kind of full extent of that chemical space and tapping into the power of novelty, diversity, and three-dimensionality for designing powerful small molecule drugs. And so what we do is focus on challenging targets. And for a particular target, we have an AI platform. So it's based on a deep learning approach that maps chemical space and finds the right region of chemical space for that target. And we then populate it with our novel molecules. And we do this based on chemistry rules curated by chemists. And this is what gives us the kind of fantastic synthesis success rate that we have, because it's not kind of pure generative AI. It's based on a lot of underlying knowledge of medicinal chemistry that we curated. So in terms of how it works, so we start with a particular therapeutic problem. We run our AI platform to identify the promising region of chemical space. We then use ChemUniverse, which is our collection of kind of medicinal chemistry knowledge, to populate it with virtual molecules. And finally, we use a multi-parameter optimization approach in order to get the right balance of properties for the molecules at hand. So we select some virtual molecules, usually quite a small number, actually. So we tend to select kind of 20 to 30 molecules. We then go ahead and synthesize these and test them, and the results get fed back into the platform. So we do kind of cycles of this for different projects. As starting data, we use any available ligand data, structural data, things like kind of fragment screening and DEL and so on can make a good starting point for our platform as well. So we can kind of work alongside those. So we take all the data we have around the target, and then we use that. And typically in the first screen, we're focused on getting as much diversity as possible into the molecules and testing different hypotheses. And then kind of from the second screen onwards, we'll be focusing in on a few prioritized kind of series and really exploring the chemical space around those. And what's really unique about the company is so ChemUniverse is our proprietary repository of medicinal chemistry knowledge. It's been curated by chemists over a period of years. And this is a collection of medicinal chemistry kind of rules and strategies, and this is what we use to populate our chemical space and generate our molecules. And then finally, our platform delivers novel and IP-free chemical space. So we check for kind of existing patents within Reaxys and other databases to make sure that any molecules we deliver are novel and have a strong IP position. So in terms of where we are compared to kind of other people's chemical spaces, so in this graph, so Chem Universe is our chemical space, and we kind of compared our molecules to Inamine and Kuehl, Campbell, and Sure Campbell. And we looked at two things. So we looked at the novelty of the molecules, which is kind of how many molecules, what fraction of molecules from each of these libraries was similar to the other ones. And then we also looked at diversity within the library, which was a measure of the different molecules, how different they are to one another. So Campbell and Sure Campbell are kind of, they're kind of like known chemical space. So Sure Campbell is the subset of Campbell that's just patents. MQ is an aggregator, so you wouldn't expect it to have a lot of novelty compared to the others because it's essentially a kind of superset of them. So these are kind of low novelty for that reason. Inamine Real is really widely used in the industry, and they have a high novelty and are really focused on that. But where we fit is, so Inamine Real has a lot of novelty, but not necessarily a lot of diversity within the library because they focus on having kind of 1 or 2 synthesis steps and building things around that. We have a few more synthesis steps, so we're able to kind of really branch out into new areas of chemical space and deliver on things where chemical novelty and diversity are a particular key factor for success. And I'm just going to show you a few of our projects and the data from those as well in order to illustrate how we work in practice. So this was a case study on a kind of hit ID against a CNS GPCR target with selectivity challenges. So we do a lot of CNS projects because the ability to kind of customize the properties, including things like crossing the blood-brain barrier, is really helpful there. And also selectivity is often a really key challenge there as well. So in this particular case, the GPCR in question had a lot of data around it. So a lot of ligand data and structural data. The selectivity was really an issue, and also kind of getting out of the known chemical space in order to overcome a certain set of ADME properties as well. So that was also something that we focused on. And in terms of the results, so for the first round of predictions, so for the hit discovery, we selected 35 molecules. We're able to synthesize 31 of them, so 89% synthesis success rate, which was fantastic. And then out of that set, we built in a lot of diversity within those molecules. So out of the 31, we were able to identify 10 active molecules across 6 different chemical series, which was fantastic. And also we were able to get nanomolar potency straight away with the 10-fold selectivity window, which was really promising because selectivity was one of the key features we were looking for. Even though it was a very kind of busy space in terms of competition, our molecules were really different from anything else that's out there. So this kind of top graph here shows the Tanimoto similarity to other known competitors. So the median Tanimoto similarity is 0.24, which is really low. And also in terms of complexity, so our molecules are quite complex for small molecules. A lot of the other molecules shown here are kind of peptides and macrocycles and things like that. So we're kind of delivering the complexity of those, but in the form of a small molecule. So we then went through a second cycle, and the main purpose of this was to improve the selectivity while maintaining the potency. So here we had kind of more molecules we really wanted to explore kind of the chemical space around the active series. So we selected 101 molecules, synthesized 83 of them. And this is still ongoing work, but out of the molecules that is tested so far, we've got 34 actives, and we're able to improve the selectivity window to 70-fold, which was fantastic, while still retaining that really good nanomolar potency, which we're very happy with. So this is still an ongoing project with a partner. And then I wanted to show you a second example, which was more around kind of how do we work in situations where not a lot of data is available. So this is another GPCR, but the challenge here was very different, which was that there was very little work done on it. There was a real kind of data scarcity challenge, no structural data at all. We had one ligand series that was known for that target. So in this case, we did basically, we did extensive homology modeling. So we came up with a really good model to enable the structure-based drug design. The exciting thing about this project was that the collaborator came to us because they tried lots of other kind of hit ID kind of methods and weren't really able to generate anything with kind of particularly good activity. So this is why they worked with us and were able to deliver some really nice results for that. So in terms of what we got here, so we selected 13 molecules. We're able to synthesize 12 of them, so 92% success rate. And out of that, we actually got 5 active molecules across 3 different chemical series, which was fantastic. And they were really delighted with that because really nothing else had worked up to that point. There wasn't a lot of kind of competition to compare it to, but compared to the one no ligand series, again, our molecules are very different. So very low tiny motor similarity there, and also kind of quite complex for a small molecule again, which you can see in this graph. So in this first cycle, we were able to get kind of low micromolar potency. We then did a second cycle of predictions where we prioritized 2 of the series from the initial screen. We selected 35 molecules. We're able to synthesize 31 of them. And out of this, we found 26 active molecules. And the nice thing was that here we were able to get from the micromolar to the nanomolar kind of potency range, and also achieve the desired kinetic profile for these. And this is at the stage where the molecules are hopefully about to be licensed. So that's where we are with it. So just in summary, we specialize in small molecule drug design. We're very much a kind of medchem specialist. We consistently deliver chemical novelty. We build in kind of complexity and 3-dimensionality as appropriate for the needs of the target. So we focus on kind of sp3-rich molecules, and we focus on chemical diversity as well. So really kind of getting out there in chemical space and coming up with new possibilities for drug discovery, while still retaining this kind of fantastic synthetic accessibility. And I'd be very interested to talk to people about any kind of opportunities to collaborate on exciting targets together. So thank you very much for your time.
Thank you very much, Helena. That was brilliant. Do we have any questions for Helena?
Yeah. Hi, Elena. Thanks, and also congratulate for your latest funding success.
Thank you very much.
Great news. Yeah, my question is, you showed 2 case studies. Those are your own pipeline, or it's third party you provided the service?
Sure. So one of them was actually from our partnership with SiSaf. So
Okay.
this is a joint project we've had for a while. And then the second one was with an external collaborator as well. So these are kind of external industry projects rather than our own pipeline.
Okay. Thank you.
Thank you. Do we have a couple of minutes in case anybody else has any questions? I can't see anything in the chat box, but feel free to jump on and come off mic.
Thank you very much.
Wonderful. Well, thank you very much again, Helena. So I will just spotlight our next speaker. We have Suzanne Safi-Siebert from SiSaf.
Yes.
Oh, there we go. I can hear you now.
And can you see the screen?
Yeah, we can see the screen.
Fantastic. Okay, thank you very much for having me today. Well, I think we are just 1 minute before noon, so good afternoon. My name is Suzanne Safi-Siebert. I'm a CEO of SiSaf, a drug development company based in the UK. In the next 10-15 minutes, I would like to share with you how SiSaf is delivering on the promises of the RNA therapeutic. Okay. First thing first, here are forward-looking statements as part of good housekeeping. And right. So we develop RNA-based therapeutic in area of the high unmet medical need using our proprietary delivery technology. This technology stabilizes both RNA and the carrier system, and it offers enhanced targeting, localization, and higher safety of the products. Our current focus is on rare autosomal dominant diseases, but we also have discovery programs in areas such as pancreatic cancer. Both our pipeline and our enabling biocourier technology are protected by a comprehensive patent estate. We are in process of raising $50 million to fund clinical trial and to expand our pipeline. Our plan, or at least our aim, is to IPO in the next 3 years, and also we are looking for a merge with a big player in order to make the most of the potential of BioCourier technology. We are a very small UK company, and with that sort of technology, it's always good to have a bigger player. So let me start with our lead program. This is for the treatment of the rare skeletal disorder, osteopetrosis ADO2. This condition doesn't just affect bone density, but it can also lead to the blindness, hearing loss in children. There is currently no treatment or cure for osteopetrosis, and SiSaf siRNA-based treatment will be the first and actually only treatment as of today for this debilitating condition. We have extremely promising preclinical data demonstrating that our siRNA formulation is able to rescue the bone phenotype and return the bone structure to normal levels. I am also very pleased to announce here in this forum that the FDA have recently granted our treatment not only orphan drug designation, but also rare pediatric disease designation. This is fantastic news for such a small company as us, permit us to pursuing a priority review voucher, and of course, expected to enter to the clinical during the next year. Our second lead program in rare bone diseases also is a siRNA-based treatment of achondroplasia, also known as a dwarfism. This condition doesn't just affect the bone growth, but also bring with it severe complication for patients, including painful spinal stenosis, sleep apnea, obesity, spinal X deviation, frequent air infections, and actually increase mortality at every age. Current treatments, the one in the market or those in clinical phase, are aimed at improve bone growth, at least marginally. And I'm stressing that the bone growth. However, they have not been shown to treat the other important aspects of the achondroplasia. One of the important aspects in this disease is proportionality. The axial skeleton and the foramen magnum has not been shown with the current treatment. SiSaf treatment target the root cause of the condition, is specific for the FGFR3 gene, and is therefore able to inhibit its signal pathway. By eliminating this mutation, we also target the comorbidities. Our siRNA treatment of the osteopetrosis and achondroplasia are currently our most advanced in-house for our in-house program. But we also have in our pipeline the partner program. This is the siRNA treatment, again, using the RNA part for the ophthalmic autosomal dominant disorder, corneal dystrophy. And also we are in the process of the proof of concept study, again, on the RNA-based therapies for cancer. So our main reason, that's at least our belief, for great success of our pipeline therapeutic in-house as well as the partner is rely on our enabling technology. Like many other scientists in the field of the drug delivery and gene therapy, I believe that RNA therapeutics to be effective, the cargo and the delivery technology need to work together in order you have a product for RNA therapeutic. And that's the key factor. Without advanced delivery technology, the ability of the RNA drug to do their job is limited to few areas, such as vaccine. I'm sure you are all aware of the lipid nanoparticles, and as we all know, LNP are currently the RNA delivery carrier of choice due to their advantages over viral-based system. Without shadow of the doubt, LNP done a great job. I believe each of us in this forum and everywhere else have the messenger RNA COVID vaccine, use the LNP for delivering of that in our muscles right now. But LNP formulations face important limitations that hold back the clinical translation of potentially life-changing RNA therapies. The key issues are limited structural integrity, which can lead to premature release of the drug, in this case, RNA. This generates a risk of the toxicity, immunogenicity, and of course, degradation of the RNA before RNA have a chance to reach to the target. This shortcoming makes application beyond vaccine difficult to achieve. Now, here's the solution. We resolve this problem by stabilizing not just cargo, in this case RNA, but also the carrier system. It is an organic-inorganic hybrid that stabilizes the organic part of the carrier system, in this case lipids. This results in much greater stability and control release, as well as significantly reduce the risk of the side effects. We have generated a large body of in vitro and in vivo data, and we are already in the clinic with one of the partner program, but on the small molecule, to demonstrate the strength of our enabling technology. This biocourier has been demonstrated able to overcome some of the major issues with the non-viral system, including instability, toxicity, as well as the immunogenicity, which currently we are facing with the lipid nanoparticles. By changing the lipids from the cationic to ionizable, by having pegylated and cholesterol, you just removing one problem, but you generating another issues. That's our true belief. As I mentioned, we generate the massive data, both in vitro and in vivo, to show better stability, also result in better tissue organ targeting. We have in vivo data demonstrating you can reach actually difficult targets such as bone tissue, chondrocytes, and that our technology can circumvent the liver, a major obstacle for many RNA treatment. Let's not forget, at least in the case of nucleic acid gene therapy, manufacturing, if it's not more problematic as the LNPs, equally have an issue here, and is a big bottleneck in order for RNA become a real product. The manufacturing process involved in biocourier formulation of RNA is much simpler and more cost-effective than with LNP even. Unlike LNP, a biocourier particle can be manufactured without RNA as part of the process and can be shipped at room temperature. And they can be loaded with RNA wherever the manufacture of the final product takes place. This factor greatly reduces the risk of the RNA degradation. This will remove the issue of the ultra cold chain, burden of the cost, and for the first time, generate global accessibility of RNA products. As I mentioned earlier, our technology is protected by a strong portfolio of patents, and that is very important as we are moving for the product. And as I'm going to share it with you, and I show you our pipeline, that technology, it has an in-house product, but also is out licensed to the partner. I did also mention we are raising a Series C, and that will really go for 3 major parts to take the osteopetrosis program to the end of phase 2, and we are planning to take the achondroplasia program to the phase 1, and also accelerate the expansion of our in-house pipeline into the cancer area. What I'm excited about isn't really about only our unique technology and the significant value it's creating. It is all about how we can change people's lives for the better. It is time, in our view, to move from just doing research to generate product using these nucleic acid and RNA therapeutic. And in order to do that, we need to think outside the box and be innovative. Our proprietary patent-protected technology generates value in 2 directions. On the one hand, is allow us to build up in-house pipeline of the RNA therapeutics, and I touch base on that. And on the other hand, is provide out-license opportunity, give a chance to drug companies to be able to make their pipeline a true drug product of future. And that's where, for us, it's really excitement aligned. And of course, I do believe we are able to make a great contribution in RNA therapeutic field, having the right technology, but with the right knowledge and dedicated team. It is our belief we are in a very promising road to success of the RNA therapeutic. That's it for me, really. Thank you very much for your attention. And you have, I believe, my email in case if you remember any question. If we don't have enough time, Jazz, and if we have a time, I'm happy to welcome a questions.
Thank you very much, Suzanne. Lots of exciting things on the horizon. We do have some time for questions. If anybody has any, please feel free to unmute, or you can pop something in the chat box. Otherwise, please feel free to reach out directly using the contact details on the screen. Okay, I think we will move on to our final presentation. So I'm delighted to hand you over now to Andrew Carnegie from ValiRx.
Perfect. Thank you very much. Thank you for the opportunity to present today. So my name is Andrew Carnegie. I'm the Head of Strategic Commercial Development in Afia Biolabs, part of the ValiRx family. Over the next 10 minutes or so, I look forward to introducing you to the company and our plans going forwards. So ValiRx is a listed company. So if you do have interest in investing, please do your own research, as we cannot be the sole source of information. So ValiRx seeks to translate academic research effectively into clinical development for new drug candidates focused on women's health and cancer. Ultimately, ValiRx seeks to unlock the vast potential of academic innovation. So how do we do that? So this is ValiRx at a glance. So the top left shows the evaluation projects. This is an ongoing process. So as a team, we talk to biotechs with early stage compounds and university groups. And if they're interested in out-licensing their products to us, we will bring them in for an assessment, which is a batch of assays. And if they operate in our hands as they do with the university group or the biotech, we'll in-license them and bring them in. This is very early stage projects, which is quite different to how early most companies will consider candidates. And then we'll take them through preclinical and out license just before the clinic. And moving on to the next box, we have therapies undergoing that out licensing process currently. We also have Cytolytics, which is a single asset preclinical subsidiary. This is based around a triple negative breast cancer molecule. And then we're also developing a strategy around our specialist translational CRO. This has been initiated with a lab build in Nottingham, Medicity, which is in Afia Biolabs. And across these areas, we have a focus on oncology and women's health. And this is all driven by our highly experienced management team. So the ValiRx company strategy, you can see from the cartoon at the base, as I've mentioned, we start at early discovery, where we do that assessment, that range of assays on potential candidates that we're looking at in licensing. And if they pass our test, we will bring those projects into ValiRx and we will take them through the preclinical process. We do that we provide the scientific, financial and commercial framework to enable that rapid translation of the innovative science through that preclinical process to the clinic. And then we just out-license it just before the clinic. So just before going into the clinic, we will out-license the molecules. For our scientific focus, so as I mentioned, we have CLX-001, which is our triple negative breast cancer molecule, which is in our subsidiary Cytolytics. We then have a relationship with Barcelona University, which is under an evaluation agreement. We have a couple of other products in the preclinical process. And then we have some legacy molecules, which is VAL201 and VAL401 for prostate cancer and lung pancreatic cancer. They are in phase 1 and phase 2. But as I said, our new strategy is to out-license just before the clinic. So they are our legacy molecules. So why are we setting up a TCRO? Well, only around 10% of academic drug development is commercialized. So you have the grant funded stage, but then there's this valley of death around these niche areas. So that's around chemical synthesis, stability, through to pharmacokinetics, formulation, regulatory, trial design and toxicology. So we're looking at bringing those niche capabilities together, either via acquisitions or by partnering up with other companies to really form this solid block here to help navigate that valley of death. So that will either be killing compounds early, so they're less cost effective and time draining as you try and get those molecules to the clinic. So killing it early is also a success. And we will also help decide which molecule should really move forward into the clinic with the most success. So here you can see that the strategy. So a traditional CRO will have data generation and core infrastructure, and we will build on top of that with bioinformatics and AI and data application. So that Afia will stand out while bringing these other blocks into play. So a part of the strategy as well is we are registered on scientist.com, so our services are now on there. We found that some biotechs and pharma companies will only purchase services and products through scientist.com, so it's very important for us to be on there. And as you know, they're a world-leading AI-powered R&D marketplace. And we also have started these collaborations. As I mentioned, that's the plan to really navigate that valley of death. So Physiomics is a leading oncology consultancy using state-of-the-art virtual tumor technology and other tools to predict and better understand the effects of cancer treatments. We've also collaborated with Oncobone, which is a third-party project management and communication company specializing in oncology. So it's a great fit for us as well as Physiomics. And Oncobone connects pharma and biotechs to service providers. So our capabilities really fit in their platform, and they're looking at having an all under one roof capability, and we really fit in with what they were looking for. So just an introduction to Afia to build on what I've said already. So as I mentioned, we're based in Medicity in Nottingham, and we're focused on both standard and specialized cell-based technologies. So our capabilities are based around efficacy and selectivity assays, mechanistic and binding assays, and cell markers and hormone measurements. And our plans for the future include cell culture assays and more complex 3D models, live cell imaging and direct binding measurements, and conducting in vitro and ex vivo testing. We're also very proud to announce that we purchased the assets from Imogen Therapeutics, and this includes a biobank of patient-derived cells, a large source of laboratory equipment, which will increase our capabilities in the assays we can offer, and also data from Imogen's in-house screening program. We're also very pleased to be able to share that Gareth Griffiths has come on as a scientific advisor. He was the Imogen CSO. So this acquisition will provide more relevant cancer cell models for cell assay screening, and also enable us development of personalized cancer models. So a little bit more about the Physiomics collaboration. This will bring biological modeling, data handling and visualization, advanced statistical analysis, and machine learning techniques. So our core team is made up of Dr. Suzanne Dilley, who's our Chief Executive Officer. She's an entrepreneur, a pharmaceutical scientist by background with translational expertise, and also a chemistry PhD. And then we have Dr. Kevin Cox, our non-exec chairman. He is a serial scaler at Pharma Services and over 25 years in life science leadership with M&A experience in manufacturing, vaccines, lab services, and biomedical imaging. And then we have Dr. Kathy Triller-Stewart, our Chief Scientific Officer. She's a pharmacologist with over 25 years in life science leadership across academic, startup, biotech, and big pharma experience. And she has taken multiple candidates through to market. So Dr. Susan Dilley and Dr. Kathy Triller-Stewart form our background of that scientific knowledge that I shared earlier, as well as the rest of the team that's part of our family now. And then we have Mark Trahan, our corporate development manager. He has a network of innovators and relations with investors that build on that business development experience that he has. So just to re-summarize, so ValiRx at a glance, as we have those evaluation projects where we're talking to biotechs and university groups about their molecules and whether they're looking at out-licensing them to us, and they'll go through that internal assessment, and then we'll in-license, take them through to the clinic. We have those therapies undergoing out-licensing, and that just before that clinic stage. And then we have cytolytics built around our triple-negative breast cancer molecule. And then we have that specialized TCRO strategy that I mentioned that's been initiated with that lab building Nottingham. And we're expanding on that with some collaborations that I've announced during the presentation as well. And that's all focused around oncology and women's health and driven by that highly experienced management team. So please reach out to me if you're interested in discussing any of the cell-based assay experiments in the oncology and women's health space that I've mentioned. If you have a technology or cell assay or model you think may be of interest to us for bringing on board into our lab, or if you're interested in collaborating, you can contact me directly, or please chat to me during a networking event. I'm often at the One Nucleus events, or pop by our booth at a future event as well. So there, my contact details are on the screen, and thank you for listening to the presentation today. And there's just a nice picture of the team from a recent meetup that we had. So thank you very much for your time, and open to questions.
Thank you, Andrew. That was great. So we do have some time for questions. And whilst some of you might be thinking of some questions or some comments, it's worth highlighting that Andrew and also Helena will be out on Helix on Thursday. So yeah, please do go and grab them if you have any questions, and yeah, if you want to have those conversations in person.
Andrew, thanks for that clear presentation. Could you please elaborate on the rationale for doing your out-licensing just before first-in-man studies, because you get a big increase in the valuation of the compound if you completed phase 1?
Yeah, I think it's just a focus for the team. That would be more of a question for our CEO or CSO. But I know it's a strategy of the company. We did used to take candidates into the clinic, as I showed on the slide, but it's now a new strategy that we brought on, where we are just out licensing just before the clinic. And I think that's come down from the kind of board level, really, Alan.
Okay, all right, fair enough. Thanks.
Thank you, Alan. Do we have any other questions from the audience? Wonderful. Well, I think we will wrap that session up there. Thank you so much to all of our speakers today, and thank you to everyone for attending. I hope you enjoyed the session, and please do feel free to reach out to any of our speakers via the app. Don't forget to vote as well. We will be back tomorrow at 10 a.m. for another innovation workshop with Pharmenable, so we hope to see you all there. And if you will be attending on Thursday, we are looking forward to it, and again looking forward to seeing you there. So thank you so much, everybody, and I hope you enjoy the rest of the conference.
Thank you.
Thank you. Bye.